Dear children, the first chapter of 8th standard biology is Life's Mysteries in Little Chambers. In this chapter, we are going to study about cells, especially plant cell and animal cell. Without any delay, let's go to the chapter. Don't you know that the body of all organisms are made up of cells? Of course you know that. Cells are the structural and functional unit of an organism. Whether it is an animal or plant, its body is made up of cells. Is there any way to observe the cells? Of course, microscope. We can use microscope to view the cells. What is the function of a microscope? The function of a microscope is to magnify the objects. So we can see a macro-sized images by using a microscope. Microscopes in which more than one lens is used are called compound microscope. In the first picture, it is a compound microscope, whereas in the second one is a simple microscope because only a single lens is used in a simple microscope, but in the case of a compound microscope, more than one lens is used. See the parts of a compound microscope. There is an eyepiece at the top and knobs to adjust, and there are objective lenses, and below that we can see stage and clip. Here we place our specimen, and below that there is a condenser. At the basal portion we can see a mirror. So remember, this mirror has two sides or two planes. One side is a plane mirror which is used to reflect the sunlight, but the other side is a concave mirror. This mirror is used to reflect artificial light. So remember the names of a compound microscope: eyepiece, knob, objective lens, stage and clip, condenser and mirror. Why is a mirror fixed in a microscope? The mirror is used to reflect the light. Plane mirror reflects sunlight and the concave mirror reflects artificial light. Why do we use a slide made of glass to place the material to be observed? The light needs to pass through the specimen, then only we can observe it. So the slide should be transparent. That is why we use glass slides. Now let's see the steps in the preparation of an observation material. In the stage 1, put the material to be observed in the petri dish containing water to prevent dehydration. If we place the material in water, we can prevent the loss of water, that is, prevent dehydration. Then after that, we can take the material from the water and make thin cross sections of the material. And in stage 3, with a brush, shift the sections to a watch glass containing water. So in the stage 3, we can place all our cross sections in the water. Then in stage 4, put the thinnest and complete sections to a watch glass containing stain. So the perfect section is placed in a stain. Stage 5, add 1 or 2 drops of glycerin to the slide to prevent drying up of the material. So we can take a watch glass and place 1 or 2 drops of glycerin on it. After that, wash the stained section in a clean water and place it in the glycerin on the slide. Okay, so in stage 6, our section is on the glycerin. Stage 7, place a thin cover glass over the material using a needle in such a way that air bubbles does not pass. So in stage 7 we place a thin cover glass on the material and finally wipe out the excess glycerin using tissue paper. So we get a perfect slide. After that we place the slide on a microscope. We know where to place the slide. Exactly, we place the slide on the stage and fix the slide with a clip. Okay, so the light reflects through the slide and we can see the object clearly. See, this is a cross section of a plant. We can see various cells here. Similarly, look this one. We can see various types of cells. Discovering the cell. Robert Hooke was the first scientist who observed cells with the help of a simple microscope. He observed the section of cork through a microscope and called the tiny chambers seen in the section cell. So remember, the first cell was discovered by Robert Hooke. And there are some other milestones in the history of cell biology. In 1831, Robert Brown discovered the center of cell and named it the nucleus. So nucleus was discovered by Robert Brown. In 1839, M.J. Schleiden discovered that the body of a plant is made up of cells. Similarly, in 1839, Theodore Schwann discovered that the animal body is also made up of cells. Rudolf Virchow, a scientist, observed dividing cells and inferred that new cells arise only from existing cells. So children, please remember these names and their contribution. Cell theory. The cell theory was formulated by M.J. Schleiden and Theodore Schwann, summarizing the findings of various scientists. It put forward two main concepts. The first concept, the body of all organisms is made up of cells. Second one, cells are the structural and functional units of organism. So these are the two main concepts of cell theory. Remember, cell theory was formulated by M.J. Schleiden and Theodore Schwann. Now let's see the steps in the preparation of an observation material. In the stage 1, put the material to be observed in the petri dish containing water to prevent dehydration. If we place the material in water, we can prevent the loss of water, that is, prevent dehydration. Then after that, we can take the material from the water and make thin cross sections of the material. And in stage 3, with a brush, shift the sections to a watch glass containing water. So in the stage 3, we can place all our cross sections in the water. Then in stage 4, put the thinnest and complete sections to a watch glass containing stain. So the perfect section is placed in a stain. Stage 5, add 1 or 2 drops of glycerin to the slide to prevent drying up of the material. So we can take a watch glass and place 1 or 2 drops of glycerin on it. After that, wash the stained section in a clean water and place it in the glycerin on the slide. Okay, so in stage 6, our section is on the glycerin. Stage 7, place a thin cover glass over the material using a needle in such a way that air bubbles does not pass. So in stage 7, we place a thin cover glass on the material and finally wipe out the excess glycerin using tissue paper. So we get a perfect slide. After that, we place this slide on a microscope. We know where to place this slide. Exactly, we place this slide on the stage and fix the slide with a clip. Okay, so the light reflects through the slide and we can see the object clearly. What are the functions performed by cells? They resist foreign particles, release energy from nutrients, synthesize biomolecules. Different parts of a cell, plant cell. You can see a plant cell here and its various parts. What are the parts you observed in the figure? Okay, let's see. Cell wall is there. Similarly, cell membrane, cytoplasm, nucleus, ribosomes, endoplasmic reticulum, Golgi complex, mitochondrion, vacuoles, chloroplast. Mitochondrion, powerhouse of the cell, helps in the production and storage of energy. That is why they are known as powerhouse of the cell. And they are abundantly seen in the cells of liver, brain and muscles, where energy requirement is very high. Endoplasmic reticulum, the passage in the cell. Conduction of materials inside the cell take place through this organelle, also known as cytoskeleton, as it provides firmness and shape to the cell. Ribosomes. Ribosomes are the center of protein synthesis in the cell, seen either attached to endoplasmic reticulum or free in the cytoplasm. Vacuole. Vacuole is covered by a characteristic membrane called tonoplast, and vacuole stores water, salts and some other excretory materials. Golgi complex collects cell secretions like enzymes, hormones, mucus, etc. in small vesicles, seen in plenty in glandular cells. Nucleus, the regulatory center of the cell. The various proteins produced in the cell play a major role in regulating various activities in the cell. The synthesis of proteins is under the control of genes in the chromatin reticulum in the nucleus. So, remember, the chromatin reticulum is seen inside the nucleus and the genes are seen on this chromatin reticulum. Hence, nucleus is considered as the regulatory center of the cell. See, this is a structure of nucleus. The outermost part is called nuclear membrane. It is a double-layered membrane that covers the nucleus. And when we analyze the nuclear membrane, we can see a large number of nuclear pores. So they are the pores in the nuclear membrane. They help in the conduction of materials to and from the nucleus. Nucleoplasm. It is the fluid part in the nucleus. Nucleolus and chromatin reticulum are seen here. Nucleolus. They are spherical bodies that play a major role in the synthesis of ribosome. Chromatin reticulum. They are seen as a network in the nucleoplasm. They carry genes, and these genes help to produce protein. Prokaryotes and eukaryotes. In the cells of bacteria, cyanobacteria and mycoplasma, no nucleus is seen. These organisms are called prokaryotes. So prokaryotes are the organisms without nucleus in the cell. But in the cells of amoeba, animals and plants, a well-defined nucleus is covered by a membrane is seen. These organisms are called eukaryote. Remember, organisms without nucleus is called prokaryotes and organisms with nucleus is called eukaryotes. Plastids are the cell organelles that are exclusively found in plant cells. These are of three kinds. Remember, there are three types of plastids and they are only present in plant cell. The first one is chromoplast. They impart color to flowers and fruits. They include xanthophyll, provide a yellow color, carotene, yellowish orange, anthocyanin, red or purple, etc. are certain pigments in the chromoplast. The second one is chloroplast. We know the photosynthesis is performed by the chloroplast. The third one is leucoplast. They are seen in plenty in the cells where food materials are stored. As fruits ripen, chloroplast changes to chromoplast. Starch is converted to sugar. This is the secret behind the change in the color and taste of fruits. So during ripening, the chloroplast changes to chromoplast. Okay. Observe the illustration showing various cell organelles in the animal cell. We have already seen plant cell and its cell organelles. So now here we are studying about the animal cell. So the outermost part is cell membrane. Remember, the cell wall is absent here. There are two additional parts in an animal cell. First one is centrosome. Centrosome is the cell organelle that is found only in animal cells. The centrioles that play a major role in the cell division are seen in the centrosome. Remember, centrioles are seen in centrosome. Centrosomes are only found in animal cell, which means centrosomes are absent in plant cell. The second one is lysosome. Lysosome contains the digestive enzymes that are required for the destruction of foreign substances entering the cell. It is seen in animal cells. Centrosomes and lysosomes are only seen in animal cell. Cell organelles which is found only in animal cells include centrosome, lysosome, etc. Similarly, plant cells include cell wall, vacuole and plastids. And these components are absent in animal cell. And there are some other cell organelles. They are found in both animal cell and plant cells. They include nucleus, cell membrane, then ribosome, mitochondria, endoplasmic reticulum, etc. So please remember this diagram. Thank you.